Hello, everyone. Now let's start the chapter 5, virus structure and multiplication. In this section, we are going to learn uh, the meaning of the virus. So virus infect every type of cell. Okay, include the precursors such as the bacteria. um, Also, the eukaryotic cells, such algae, fungi, protozoa, plants, and animal humans. So, so there are tons of viruses in the world. For example, just 1 milliliter of sea water contains 10 million viruses. Okay. Um, However, so the virus has existed in the world for many years, billion years. However, the human the human has the human has known the virus since a very short time. Okay, since the Even Hawke invented the scope to visualize bacteria. Then the scientists know that bacteria can cause diseases, can cause infection. However, for some cases, the physician or the scientists found that even they cannot find the pathogen from the specimen of the patients. Okay, so there were no bacteria. However, the patient still have the symptoms. So so this is the question that. So this is the question. So this is the stuff for question that so the scientists cannot find the answer for a long time. So the Louis Pasteur has the hypothesis that there are some living organisms that much smaller than the bacteria that cause diseases. So one Louis Pasteur studied rabbits. So he postulated the term virus for the things that he hypothesizes. Okay, so the virus is the Latin word means poison. Then later on, Ivanovsky and Bejerick showed that a disease in tobacco was caused by virus. So the Loeffler and French discovered the animal virus that cause foot and mouth diseases in cattle. So so this is another evidence that scientists believe that virus can cause the infection, because if the scientists filter the food containing the living organism, so the filtrated can still get the infections. Okay, so that means there are some pathogens that smaller than bacteria that can cause infections. So, so the question about the viruses is, so are they organism or are they alive? So, what role did viruses play in the evolution of life? So, so we will answer this question. So the virus, actually, the virus are not considered as a living organism, because so the because the virus doesn't have the real life. Okay. So what is the real life mean? So what is living organism mean? So because the living organism means this organism has its own living process such as the metabolism, reproductive, okay, as the reproduction. So, for example, the living organism need to eat, drink, pee, and poop. Okay, the living organism can also make their own offspring through either sexual or unsexual reproduction. Right. However, the virus don't have this process. Okay, so the virus doesn't eat drink. Okay, virus also doesn't pee and poop. Virus also cannot so virus cannot make their offspring by themselves. So they have to infect the cells and use the machinery of the cells to produce their own offspring. So that's a different of virus from the living organism. So the virus, so we cannot say the virus live or die, okay, because this is not a living organism. So the virus is better. The viruses are better described as active or inactive, okay. So since the virus have to invade the cells to survive or get or reproduce their own offspring, so the so once the virus is invaded the cells, so they can change the so they can change the cells gen, so the genetic makeup. So they can also change the cells functions. Okay, they can also cause the cells tissues die. Also, once the virus invades the cells, so some viruses will conjugate their genome to the host genome. So, for example, 8% of human genome has been found from the viruses. Okay. So 10 to 20% of bacteria genome contains the viral sequence. Actually, so the viruses are considered as the obligate intracellular parasites, okay, because they need so they need to survive in the host cell. They need the organelle of the cell to make to reproduce to make the new viruses. So the viruses are obligate intracellular parasites of precursors and eukaryotes. So there are tons of viruses in the earth, okay, on Earth. So they are everywhere, okay. So they are in the water, in the soil, in the air. So the viruses are very small, are much smaller than the cells. So the range is from 20 nanometer to 1,000 nanometer. So the cells, viruses are not considered as the cells because so their structure is different from the cellular structure. So their structure is very compactable and is very simple. So the viruses does not have viruses have their own independent life. So they need So their life is dependent on the cells, on the host cells. So the structure of the viruses is the protein shell and the nucleic acid in the protein cell, protein shell. So the nucleic acid contains all the genetic information for the viruses. The nucleic acid can be the DNA or RNA, but not both. So based on the nucleic acid, so we can categorize, so we can classify the viruses to the DNA virus or the RNA virus. So the nucleic acid can be the double-strand DNA, single-strand DNA, or single-strand RNA or double-strand RNA. So on the surface of the protein shell, so there are many molecules. So those molecules can be used by the virus to attach the host cell, to invade the host cell. So normally the viruses doesn't have enzyme for the metabolic process or for the reproductive process. So they need to borrow the cluster of enzyme from the host cell to perform their own metabolic process and the reproductive process. So how we name the viruses? So we can name the viruses based on their host and the diseases they caused. Okay. So now we have taxonomy of viruses based on the host and diseases that they cause, the structure, the chemical the chemical composition, and the similarity in the genetic makeup. Okay. So based on this system of so based on this named system, so we can classify 8 orders and 38 families. Now let's work on this question. Which of these following best describe viruses? So I'll give you 5 seconds so you can answer this question. Okay, so we can see the heterotrophic. Okay, heterotrophic. Actually, it's not correct. Okay, so actually, so the viruses can only so the viruses don't eat and drink. Okay, so this means they are not heterotrophic. Okay, so they only invade the cells to get their own life. Okay, saprobic is not true. Okay, because the viruses invaded the live cells for their metabolic and reproductive process. Chemoautotrophic is not true because so the viruses cannot make their own food based from the chemicals. This one is not this one for the plants or the grass. So the C is correct. So the viruses have to invade the cells. Okay, invade the cells. So this is obligate intracellular parasites. Now let's work on the section 2. Let's talk about the structure and function of viruses. So the viruses are the smallest infections particles. Okay. So the smallest one is parvovirus. is about 20 nanometers. So the largest one is the herpes. Herpes simplex virus is 150 nanometers in mass. Some cylindric viruses can be 800 nanometers, but it's narrow. It's 15 nanometers in diameter. Okay. So since the viruses are very tiny, so we normally we cannot visualize the viruses with the microscope. We have to use the electron microscope. Okay. So this diagram shows the size of viruses compared to the eukaryotic cells, the yeast. So we can see the viruses are very tiny. So the viruses have many different shape and size. Okay. So the virus, so the structure of virus is very simple. Viruses don't have their own enzyme system to reproduce the new viruses. Also, they don't have the raw materials such as animal acids and sugars for making new viruses. So the virus structure contains regular repeating subunits, so that gives their own crystalline appearance. So the structure also contains the parts required for invading and control the host cells, such as external coding. Okay, external coding. And normally, it's a cellular membrane pitching from the host cells. Also, protein shell. Okay, and the inside the protein shell, so that's nucleic acid. So that could be either DNA or RNA. Some viruses can carry one or two enzymes. Okay, however, not a lot. So they still need the enzyme system from a host cell. Now let's talk about the protein shell. So the protein shell is also called capsid. So since the protein shell surrounds the nucleoside, so the capsid is also called nuclear capsid. Okay, because the capsid surrounds the nucleic acid. So the capsid together with the nucleic acid is called a nuclear capsid. Naked viruses consist only of the nuclear capsid. So the viruses also have envelope. So the envelope is the external covering of capsid. Normally, so this is a piece of hosting cells membrane. So either from cellular membrane or the nucleus membrane or the or the membrane from an ER system. So the spikes can be found on some viruses environment on the surface of on the surface of the environmental viruses. So spikes are the projection from the nuclear capsid of the envelope. So the viruses can use spikes to to attach the host cell and dock with the host cell. So the one matured or formed viruses. So if this so if this virus has the capacity to invade the host cell, so it is also called a variant. Okay, variant. For example, this is variant. This is also virus. Okay. So this is capsid. Okay, this is capsid. So this is a nucleic acid. Okay, this is a spike. So the virus can also pinch a piece of host cell's membrane on the surface, so that could be the so that will be the envelope. Now let's talk about the capsid. So the capsid is made from proteins subunits. So the proteins subunits is called capsomeres. So these capsomeres can be assembled to the capsid. So there are two different types, helical and icosahedral. Okay, icosahedral means so this is a structure with 20 surface. Okay. Enveloped viruses. So these type of viruses have to pinch a piece of cellular membrane on their surface. So that piece of armor can protect the viruses. Also, normally, so the virus get this piece of cellular membrane from the cellular membrane, nuclear membrane of the endoplasmic reticulum. Since this virus is covered by the cellular membrane, so the envelope will make the virus very flexible shape. Okay, so it can have a different type of shape compared to the naked virus. So here we can see the helical capsid structure. So the helical capsid. So this is round. This is a rolled shaped capsomere that bound together to form a series of hollow discs. So the naked virus, for the naked virus, so the capsid are very rigid and tightly wounded to cylinder-shaped package. So for the enveloped helical nuclear capsid, so are more flexible and tend to be arranged as a loose helix within the envelope. So this can be found in several human viruses such as influenza, measles, and rabies. Icosahedral capsid. Icosahedral capsid. So the capsid is formed with 20 sided surface. So the shape can vary from one to another. For the naked viruses, such adenovirus, the example of the naked isosaccharovirus. So we can see they are so they have different so they have 20 surface, okay, 20 surface. Also, this type of virus also contains a spike, okay, so this is a spike. So the envelope, if the virus is covered by the envelope, so their shape can be changed. So the shape can be changed. So we cannot see the classic shape. We cannot see the classic shape as the naked capsid. So so the virus can also have the complex capsid structure. So the complex capsid, that means so this capsid is made of the conjugation of the many type of proteins. For example, they can have the capsid head. Okay, they also can have the protein sheaths and protein collars or the terpenes. Okay, so the good example would be the bacteriophage. Bacteriophage have this specific structure. Now let's talk about the genome. Okay, so the genome means all the genetic information carried by the organism. So for the virus, so their genome is very small, is very simple, because since the virus is very tiny, so they don't need too much information. For example, there are only four genes for the hepatitis B virus. For the herpes may have more. They have hundreds. However, compared to the eukaryotic cells, so hundreds is still less. Okay. So virus only carry the genes they need for the invading the host cells or controlling the hosting cells. So based on the chemical components of the virus, we can classify the virus as the DNA virus and RNA virus. So the DNA virus means this virus contains DNA. So that could be either single-strand DNA or double-strand DNA. RNA means this virus only contains RNA. So that could be double-strand or either double-strand or single-strand. Positive sense RNA means the direction of the RNA is from the 5' to 3'. It's ready for the immediate translation. Negative sense means the direction is from 3' to 5'. So so this direction had to be converted to the positive sense before the translation. Segmented RNA means the genes exist on separate pieces of RNA. Retroviruses actually, so this is the RNA virus. So they carries the specific enzyme can convert RNA to DNA. Then use the DNA so they can translate the DNA information to make the proteins for the new virus. As we can see from these tables, so this is DNA virus, double-strand DNA could be the smallpox. Single-strand DNA could be the erythema infectiosum. RNA virus could be the poliovirus, influenza virus. Influenza virus is the negative sense. So the retrovirus is double-strand RNA. HIV is single-strand positive, reverse transcription, so the retrovirus, HIV is the retrovirus because they contains reverse transcriptase. So the enzyme for some virus needs the enzyme from a host cell, from the host cell to make the new virus. So the enzyme they need would be the polymerase. So that can synthesize DNA and RNA. Okay. So replicates for copying RNA, and the reverse transcriptase synthesize the DNA from the RNA. So the virus doesn't have the genes for the metabolic enzymes. So the viruses also don't have the raw materials to make the new viruses. So that's why they need the machinery from the host cells. For example, they need the ribosome. Okay. They also need the tRNA from the host cells. So now let's work on this question. Which of the following is not a type of viral nucleic acid? So I give you 5 seconds. So we can see for A, yeah. So this is true. Some virus is the single-strand DNA. Some virus is double-strand DNA. Double-strand RNA. Some strands is double-strand DNA. Some strands so some virus has the segmented RNA. So that's true. So the correct answer is E. So all of the types found. So that means all these can be found in virus. So that's not true because so the virus can only has one of these. So now let's study section 3. So the life cycle of the virus. Since the virus are the parasites, so they need to invade the cell and control the cell. So the viral replication cycle is dependent on the way the virus is transmitted. Okay, how they replicated in the host cells, and how they respond to the immune system of the host cells to avoid being killed. So the phases of the virus invading and replication cycle include adsorption, penetration, encoding, synthesis, assembly, and release. So the length of the replication cycle varies from 8 hours to 36 hours. So the virus can specifically invade one or certain host cell. So virus cannot invade every cell. That's because so on the surface of virus, so there are specific spikes and molecules. So those spikes and molecules can recognize the specific receptors on the host cells. So if the host cell doesn't have the receptors for this virus, so the virus cannot invade this type of cell. So the host range, so this limited range of the cells that the virus can infect. For example, hepatitis B can only infect liver cells because the liver cells has receptors for the hepatitis B virus. So the hepatitis B virus cannot infect the heart cells, the muscle cells, the nerve cells. No, it cannot recognize those cells. Poliovirus can infect intestinal and nervous cells. Rabies can infect several cells of the animals. So if the cell doesn't have the virus receptors, so they are resistant to the invasion of the virus. So the specificity of the virus for certain tissues is called tropism. So as we can see this diagram, so first of all, so the cell has to have the receptors for the virus. So the virus can use their spike to combine with the receptors and then invade the cell. Once the virus recognize the molecular receptors on the cells, so they can invade, so they can penetrate into the cell. So when the virus penetrates the cell, so the cell membrane can make a small package to totally surrounding the virus, to make a vacuole. So this is called endocytosis. So the entire virus is engulfed by the cell and to form a vacuole. And then so the virus viral envelope can fuse with the host cell membrane, because so the envelope actually is so the envelope actually comes from the host cell membrane, so the structure and chemical components are exactly the same as the host cell membrane, so they can fuse together. Once the envelope fuses together, so you can release the nucleic capsid to the cytoplasm of the cell to release the nucleic acid of the virus. as we can see here. Once the virus combine with the receptors of the cells, so they can so the endocytosis can happen. So the entire viruses can be engulfed into the cell, and then so the viral envelope fused with cells and then release the nucleic acid. Now let's talk about the DNA viruses. DNA viruses, because this type of virus contains DNA, so they can release their DNA to the host cell's nucleus and release the DNA. The DNA can be replicated and make more DNA. R virus. So the R virus. This type of virus will invade the cytoplasm of the host cell. So the RNA can be directly used by the ribosome of the host cell to produce the components of the viruses and to make the new viruses. Therefore, the retroviruses, so they need a reverse transcription. So they need so this type of process happen in the cytoplasm. So they need convert the RNA to the DNA in the cytoplasm, and then DNA will get into the nucleus to make the new viruses. Now let's talk about the life cycle of the double-strand DNA viruses. So for the early phase, so the viral DNA get into the nucleus. Because in the nucleus, so the DNA is transcribed to the messenger RNA. So the viral DNA can be transcribed to the messenger RNA. Then the messenger RNA can migrate from nucleus to the cytoplasm to attach to the ribosome to translate the viral information and to make the new viral protein. Also, so the host cells can also make more new viral DNA in the nucleus. So the DNA polymerase is involved in this process to generate more viral DNA. So the later phase, so the part of the viral genome are transcribed and translated to the protein. So in the later phase, so the protein are generated. So those proteins can be used to form the capsid and the other structures. And also in this phase, since the DNA is generated and protein is generated, so DNA and the proteins can be assembled to form the new virus. Once the new virus are assembled, so they can be released from the host cell by budding or the cell's disintegration. So the assembly Assembly means we so the host cell insert the viral DNA to the capsid. So release means the virus is released out from the host cell. So the release can be controlled by the size of virus or the health of the host cell. For example, so the pox virus infect cells can make 3,000 to 4,000 vision, and the poppy virus infected cell can make 10,000 vision. So this is the release of the virus. So the new virus can form the bud. So the bud can form the bud. So the bud can pinch a piece of cellular membrane as their own envelope. So once again, let's review the life cycle of the viruses. So the first is adsorption. So the adsorption, that means the virus recognizes the host cell and recognize the receptor on the cellular surface for the virus. So the virus can combine with the receptors. So this combination could be regulated by the glycoprotein spike. So this spike will recognize the receptors for the virus on the surface of host cell. Once this recognition is done, so the virus can get into the cell through by the process of endocytosis. So the cell will make a vacuum and surrounding the entire virus. It's like the virus is engulfed into the cell. And then, so the host cell can digest the viral envelope and capsid. So this is called uncoating. So the nucleic acid can be released to the cytoplasm of the cell. And then, so this synthesis for the DNA virus, so the DNA viral DNA can get into the nucleus for the transcription for the transcription and the replication. For the RNA virus, so the viral RNA can directly translated to the proteins using the ribosome in the cytoplasm. Once the DNA made once the viral DNA made a new DNA and the proteins, so the DNA and the proteins can be assembled to a new virus. So it's also the same process for the RNA virus. So the new viral RNA and the viral proteins can be assembled to new virus particles. So the new virus particles can be, and then so this is released. So the new virus particles can be released from the cell. So the release could be 2 ways for the non-envelope virus. So once the virus is matured, so they can release when the cell lysed or ruptured. However, for the envelope virus, so they can be released by the budding. Okay, budding. During this process, so the virus can pinch one piece of can pinch one piece cellular membrane from a host cells as their envelope. So once the virus invades the host cell, actually, so they can cause the change of the change. So the function change of the host cell is also called as the cytopathic effect. Okay, this means the virus can induce damage to the cell. It can change the cell function. So the types of the CPEs include the growth change of the shape and size. So we can visualize this through the microscope. The second is the change the structure in the cellular. Okay, so in the cells change or the. So the third is the inclusion bodies. Okay, so the cells generate new structures. So actually, so the inclusion bodies are the damaged organelles. Okay, damaged organelles. So they change their morphology, change their shape and size. So it's look like the new structure. Synthesia means the infected cells will fuse together. So their cellular membrane will be broken and fused together to make a new and big cell. So this new and big cells, we can see multiple nuclei in this new and big cell, because several cells fuse together. So the nuclei will flow in the cytoplasm of this germ cell. So this is called synthesia. So the synthesia actually is a type of dead cell. So this cell doesn't have any functions. So eventually, so the virus will kill the cells. So as what we can see in this pictures, so this is synthesia. Okay, this is synthesia. So this is a huge cell compared to this one. It's normal cells. So this is inclusion body. So we cannot see this body in the normal cells. However, so the virus, so once the virus infects cells, so for sometimes cases, so the virus will not cause the significant damage to the cell. So the cell can still be alive. Okay, so the cell will live with the virus in their body. So this can last for weeks or years. So it can be latent to the cytoplasm. So provirus is the viral DNA incorporated to the host DNA. Okay, and it's like measles. So that means the viral DNA will be conjugated to the host DNA. Okay, and once the host cells replicate and transcript the DNA, so the virus DNA can be replicated and transcribed as there. And then another is the chronic latent states. So this is for a long time. Okay. For example, so the herpes can invade the human nervous system. So it can travel to the ganglions of the ganglion. So it can travel to the neuron in the ganglion. So it can live in the ganglion neurons for a long time. So the patient may not have symptoms. However, if the patient's immunity system, so the immunity activity is low, so the herpes can get out and cause skin rash and pain. So the virus can cause cancers. So it is estimated that 13% of the cancers can be caused by viruses. So the virus cause cancer are called as oncogenic. That means the capacity to cause cancer. So some viruses carry genes that cause cancer. It's like a papillary. It's like so we talk about this later. Some can produce the proteins and cause cancer. So how does the virus cause cancer? Because once the virus invades the cells, so it can change the cells growth rate. Also, some virus can conjugate, can incorporate their DNA to the host cells DNA, so they can change chromosomes. Some can change the cell surface molecules, okay, and some can change the cellular division. So they can change the cells. So they can change the cells properties. So the cells can divide forever. So they can make more, and so the cells can make more and more new cells. So this is the characteristics of cancer. So here are some oncovirus. So that means this virus can cause cancer, because onco means cancer. So like papilloma virus, and herpes virus. Hepatitis B can cause liver cancer. So the papilloma virus can cause cancer tumors in the genital organs, genital area. It can also cause oral cancers. So the mechanism that virus cause cancers, so we can see from this diagram. So some virus can produce certain proteins to change the cell growth, and so that the cells grows again forever, So the growth so the cellular growth is processed without any control. Also, so virus so the virus can incorporate the viral DNA to the host cell's DNA and to change the cell's function to cause the cell's growth and divide under control, without control. Now let's talk about the bacteriophage. So the phage means eat. Phage means eat. Bacteriophage means bacteria eating. Okay. So actually, this is one type of virus. So bacteriophage phage is double-stranded DNA. Most are the double-stranded DNA, but some are the RNA virus. So the bacteriophage is very specific to invade the host cells. It's very specific because for most so normally one bacteriophage only invade one type of bacteria or certain bacteria. So the most of the bacteria that invaded by the bacteriophage actually they are the pathogen for the human, so they can cause the disease for the human. So here is the example, like the bacteriophage for the E. coli. So they can infect the E. coli. Once the bacteriophage attached to the cellular membrane, so they can release the DNA, their viral DNA to the cellular membrane, into the cytoplasm to make a new bacteriophage. Again, so the attachment is called adsorption. And then, so that's the penetration. Actually, the penetration is different from the virus that we talk about. Actually, so this not the engulfing of the virus to the cell. So the viral shell is still on the surface of the bacteria. So the virus only releases their nucleic acid into the cytoplasm of the bacteria. So the nucleic acid can be replicated or transcribed by using the machinery system of the bacteria to make a new viral protein and viral nucleic acid, and then so those new viral protein and nucleic acid can be assembled to make a new bacteriophage particle. Once more and more so the assembly can make a new virus particle. So this is the variant. So once variant is matured, so more and more variants will cause bacteria lysis to release the variants. So once the bacterium is lysed, so the bacteria is die. Okay, so by this function, so the bacteria can kill bacteriophage can kill the bacteria. So the viral DNA So the temperate phages. So this is adsorption and penetration. So this is called temperate phages. Okay, temperate phages. So once the viral DNA enters an inactive prophage state, so it can insert the viral DNA to the bacterial chromosomes, copy make more and more copies of the viral DNA by the replication. So sometimes, the bacteriophage will not kill the bacteria immediately, so the bacteria can still alive. So the bacteria is infected. So the bacteria is sick. So this sick bacteria actually carry many bacteriophage DNA. So this is called lysogeny. Okay, lysogeny. So this so the lysogeny state can be last for a long time. So this can be a latent stage of the bacteriophage. And then if the environment condition or some conditions are optimal for the bacteriophage to be activated, so it can convert this stage to an active stage. So it can be activated and continuously make more and more bacteriophage and eventually cause the bacteria die. So the lysogenic conversion, so the so occasionally phage genes can cause the production of a toxin or enzyme. So those toxin enzyme can cause a disease for humans. Okay. For example, for the so this is also called lysogenic conversion. For example, for the Corynebacterium diphtheria. Okay, so it can produce diphtheria toxin. For the Vibrio cholerae, it can produce cholera toxin. Now let's work on this question. Put the phases of life cycle of animal viruses in the correct order. So I'll give you 5 seconds so we can work on that. Okay, so the first one is adsorption. Okay, first one is adsorption. So this is the process that virus encounter and recognize the cell, and then is the penetration. Once the virus attached to a cell, so they can get into the cell. So this is penetration. So the third is uncoding. So the host cell digests the viral envelope and viral shell. And the fourth is synthesis. So the viral nucleic acid can be replicated and transcribed to make a new viral nucleic acid and new viral proteins. So the five is assembly, okay, assembly. So the new viral proteins and new viral nucleic acid can be assembled to new viral particle. So last one is release. So once the viral particle is matured, so they can be released, and then the cell could be die. Now let's work on the section 4. Since the viruses are the obligate parasites, so that means we cannot use chemical medium to culture the virus, right? So we have to use living cells, so living organism to culture things. So we can do in vivo culture or the in vitro culture. Okay, the in vivo means the animals. Okay, in vivo means animals. So that means we use the animals to culture cells or the embryonic birth tissues like the chicken eggs. Okay, so the in vitro that means the so the cells or the tissues culture. So we can culture the living cells or tissues, and then inoculate the virus to the cultured cells to culture the virus. Actually, so the best way is the in vivo culture. So the why do we do the viral culture? Because we can use this method to isolate and identify the virus from the clinical specimen. Okay, so we can use the method to set up the accurate diagnosis, and then we can choose the medication for the patient. We also can isolate and culture the virus then to make the vaccine. Also for the research, so we can better understand the virus and better understand the virus structure or the genome and the so the life cycles. So we can so this is for research purpose, so we can better understand this virus, maybe find a new treatment for the virus. So the animals we use the mice, rats, or the guinea pigs, okay, or the rabbits. Where the mice is the cheapest one, so the rabbit is very expensive. So some insects and some insects can also be used the culture. Actually, however, normally we use the mammals to culture the virus. So since the virus is very host specificity, so that means so based on the host specificities, we choose what kind of animal can be used for the culture of certain virus. For the bird embryon, so we can use eggs such as the chicken eggs, duck eggs, or turkey eggs. So eggs. So the advantage of the eggs is that it's cheap. Okay, it's very cheap. Also, so the egg is closed unit. Okay, so inside, so there's a sterile environment. So it's very it's a very optimal tool to get the pure virus and isolate the virus from the specimen. Also, so the eggs also contains its own nutrition system. Okay, so we can inject the virus through the eggshells. Okay, and so the virus can be inoculated to the egg and replicated in the eggs. Then let's talk about the in vitro. For the in vitro culture, so first of all, so we have to culture the cells or tissues. So we can use the flask or disc to culture the cells or tissues. So this is the steroid devices. So once we get a monolayer of the cells on the bottom of the flask, so we can inoculate the virus in the media. Okay, so the virus can get so the virus can get adsorption to the cells. So once so in vitro cultures, we can observe the cells to know if the cells get infection by the virus or not. Because once the cells are infected by the virus, so the cells will change its shape and morphology. So we can see a plaque appears in the flask. Okay, so the plaque means area where no cells survive. Okay, because the virus infected cells so the cells infected cells are killed and float away. Because when we culture the cells, all the cells attached to the bottom of the flask. So if the cells are died, so they will flow away. So we will find an area where there are no any cells. So this is called plaques. So this is visible of the CPS. Okay, cytopenic effects. So as what we can see in this picture, so this is healthy cells. So this is the monolayer of cells attached to the bottom of the flask. So everywhere are covered with the cells. So this is the plaque. So in these areas, all the cells are died and flow away. So there are no cells. Okay, there are no cells. So these are the plaques. Now let's talk about the detection of the bacteriophage. So the virus, when the cells are died, so the virus are released. So the virus actually can release the virus can infect the other host cells, and then more and more cells will be infected and died, release more and more virus. So we will see more and more plaques. Now let's work on this question. Which of the following is not an in vivo method of culture animal virus? So I give you 5 seconds. So we can see the correct answer is C, okay? So the cell culture is in vitro. Now let's talk about no cellular infectious agents, okay? So these infectious agents are different from virus. For example, first let's talk about the prion. Actually, the prion doesn't have the nucleic acid. Okay, so the prion has only protein. Okay, so this is the protein infectious particle. So the prion can cause many diseases. Since so the human know this disease is just 10 years, okay, and so just a decades. So there are still many things that we don't know. So the prions can cause encephalopathy, okay, encephalopathy. And so actually it can deposit long protein fibril in the brain tissue to cause the disease in the brain. So one disease is Creutzfeldt-Jakob disease. Okay, so this disease was found on one Pacific islands. It's called Cruz Islands. Okay, so because scientists found this disease happened in the tribe, in the local tribe on that island, because the people in that tribe actually the people eat because so on that island, so the people eat the people. Okay, so they get this type of prion disease. So it will cause the nervous system dysfunction, and so they have the degeneration of the nervous system, and eventually, so the disease will cause death. So the ball-bone spongiform encephalopathy. So this is called mad cow disease. So we so this disease has burst out maybe 20 years ago. So this disease burst out in Britain. So it caused many cow dies. Okay. Steinbrenner syndrome. It's also called multiple system atrophy. So it's like a Parkinson's disease. So second is the satellite virus. So this is actually this type of virus actually so they have to infect the cells. So they have to work with the other virus. So they have to infect the cells that has already been infected by other cells. So this is called a satellite virus. So delta agent is the naked circle RNA. Okay, naked circle RNA. So this is naked circle RNA. So it doesn't have the protein share. Okay. So it can only express with the hepatitis B virus. So once the delta agent infects the patient with the hepatitis B, so it will eventually worsen the disease and may cause the patient die. So this is one type of the satellite virus. Virand is virus like particles that affect the plants. So it affects many economic plants such as tomatoes, potatoes, cucumbers. So it has the naked strand of RNA. So it doesn't have the capsid, it doesn't have any coding, any shear. Now let's work on this question. Crisfield Jacob disease and bovine spongiform encephalopathy are caused by prion. So which of the following best describes a prion? So I'll give you 5 seconds. So viral particles, so that's virus. No, this is not a virus. Naked DNA, no, because it doesn't have DNA. So actually it's C, okay? So this is protein. This is infectious protein. So it's not a bacterium, it's not RNA. So the virus and the human hairs, actually, so virus cause a lot of human diseases. The most common one is the cold, is the flu. Okay, so it can cause hepatitis, chickenpox, influenza, herpes, rats. Okay. So the worldwide infections would be dengue fever, yellow fever. Okay. High mortality would be the rabies, AIDS, Ebola. Some viruses infection cause disabilities like polio, rubella. Okay. Some can cause chronic infections such as type 1 diabetes and multiple sclerosis, many cancers, obesity. So treatment, actually, so all the antibodies cannot treat the virus infections. So the antibiotics cannot kill the virus. So until now, we don't have many medication to treat the virus. We do have some medication for the HIV. Okay, so those are the integrase inhibitors, so that can interrupt the HIV. So the incorporation of the HIV to the host cell's DNA. So we do have some HIV medication. So the best treatment is vaccine. So we have generated many vaccine to prevent the virus. So the virus invasions. For example, like measles, rubella. So the measles, rubella vaccine. So once we introduce those vaccine, so vaccine really dramatically decrease those type of diseases. Now let's work on this question. Antibiotics are an effective method for treating viral infections. So I'll give you 5 seconds. Okay, the question is false because antibiotics cannot kill the virus. Now, so this is the appendix, so you can use this to review all the contents. So this is end.